interested in doing research that is quite applied, because I like the idea that what we do can actually be used in the real world. So we've tended to work in areas that are of interest to the pharmaceutical industry, and what we try to do is work out how to make protein molecules that are of interest to the pharmaceutical industry, either directly as therapeutics themselves, so things like insulin would be a good example of that, or protein molecules that they need in order to develop new drugs, so these would be drug targets. So I've got a long-term interest in things called membrane proteins, which are proteins that sit in your cell membrane, and your cell membrane keeps all the things in the right place at the right time, and many of the things that have to come into and out of cells are basically mediated by membrane proteins. So membrane proteins are like windows or doors into and out of the cell through the membrane, and you can imagine, because they play such an important role in things like letting nutrients in or waste products out, or the way that cells talk to each other, then they become important parts of healthy cells. And when the proteins go wrong, that's often associated with disease. So if you can understand the proteins and what they look like, you can then design drugs that will fit directly into the protein in some way, and that can be used for therapeutic purposes in particular disease states. So what we're trying to do is, we're just interested in looking at specific proteins that are in the membrane. So for example, the pharmaceutical companies or other academic groups will have identified particular proteins in the cell that are known to have an involvement in a particular disease state. So for example, there might be some protein that's involved in diabetes, insulin resistance, that somebody's particularly interested in, or something like Parkinson's or Alzheimer's disease, for example. One of the interesting sets of proteins we're looking at the moment, in collaboration with the group at Birmingham, are involved in the way hepatitis C virus binds to cells. So these are proteins that sit in the membrane, and when the virus comes in, they act as the place where the virus can bind to the cell and then go on and do its thing. So if we can understand, for example, what these proteins that sit in the cell membrane look like and how they interact with the virus as it comes into the cell, then we should be able to perhaps block that interaction and stop the infection.
And yeast, what's the yeast connection?
So the yeast is the method to actually get the proteins in large quantities. So what we want to do is to be able to get these interesting proteins, like the components of the hepatitis C virus receptor, in large enough quantities, which for us large quantities means milligrams, which is not something you can see on a spoon, but for us gives us the ability to do the analytical techniques that we need. So we need these proteins in large enough quantities that we can then go on and do our experiments. If you just went into human cells to get the proteins that you were interested in, they're present in such tiny amounts you wouldn't actually be able to get anything useful out. So what we need to be able to do is somehow amplify the amount of protein that's there naturally. So we do that by taking out the gene from a human and putting it into a yeast. And yeast are really handy because they're like us, they're eukaryotic. And they have many of the properties that human cells have, but we can grow them to very high density, so we can make lots of protein.